So you will recall from the memorandum of Dr. Hink that I read before the break that there appears to have been some discussion at the end of the meeting about the industry as a whole not introducing anti-HBC testing. And Dr. Carr from Alpha saying that they wouldn't begin, Dr. Riddell from Armour saying they wouldn't begin unless they were forced to by a competitor doing so. You will also recall that Dr. Shrigley from Highland said he couldn't speak for his company management, and Dr. Hink gratefully parroted that line. We know that a few weeks later, on the 2nd of April 1984, Qatar publicly announced that it would commence testing all donated plasma for anti-hepatitis B core antigen. The company declared that until a specific test for any AIDS agent has been discovered, Qatar intends to take these precautionary measures to protect those with haemophilia who depend upon our products. The reference is at paragraph 198 of the written presentation. A document from May 1984 shows the debate that took place within Qatar that month when the policy was reviewed. It's a helpful document because it literally sets out the arguments for and against hepatitis B testing, and it does so in the context of an internal company document, so something not for public consumption, but for the company itself to cogitate upon before deciding whether or not to continue the policy that it had announced of anti-HBC testing. If we could have on screen, please, CGRA 40362. We can see that the memorandum is dated the 25th of May 1984, and it comes from John Hink, and it is sent to the members of a committee within Qatar, which was responsible for making decisions on matters such as these. And it refers to the materials that are being provided ahead of the meeting on the 31st of May. We turn over to the second page, please. We can see a document headed Arguments Advocating Continued Hepatitis B Core Antibody Testing. Not to give anything away, but there's on the following page, heading Arguments for Discontinuing Hepatitis B Core Antibody Testing. So the pros and the cons being set out. I'll read through this document from the top. Arguments advocating continued hepatitis B core antibody testing of Qatar plasma. 1. Exclusion of HB core antibody positive plasma for use in the manufacture of coagulation products will reduce the risk of further product withdrawals. A. National Haemophilia Foundation and FDA agree that coagulation products contaminated with plasma collected from a donor subsequently diagnosed with AIDS must be withdrawn and/or withheld from market. Until proof that the AIDS agent is inactivated by heat treatment or other means, it is unlikely that this position will be changed. B. Donor populations at greatest risk for AIDS are homosexuals, bisexuals, and intravenous drug users. C. Screening by interviews and lymphadenopathy have been effective only partially in eliminating high-risk donors. D. CDC reports 88% of homosexuals, bisexuals, and 96% of intravenous drug users with AIDS are HB core antibody positive. 78% of individuals with AIDS prodrome are positive. A high percent of populations considered at risk for AIDS have been shown to be HB surface antibody positive. We'll come back to those figures later, sir. 2. Qatar has an obligation to use all reasonable means to provide safe, effective products. A. Hepatitis B core antibody is a marker for hepatitis and an indication for exposure to multiple infectious diseases. Hepatitis B core antibody positive plasma may be expected to have greater virus contamination than untested plasma. B. Retrospective studies show 25% to 50% of hepatitis B core antibody positive blood can cause post-transmission non-A, non-B hepatitis. Coagulation products derived from hepatitis B core antibody negative plasma should transmit less non-A, non-B than those produced from conventional plasma. 3. Hepatitis B core antigen positive plasma exclusion from coagulation products should result in high customer acceptance with increased sales at appropriate prices. A. Qatar, without chip data, needs to differentiate itself from the other manufacturers to gain back market share. B. If HTLV screening test is available at year's end 1984, Qatar will have 8 months production from core screened plasma available before any HTLV3 screen products are available. C. If HTLV3 screen test is delayed, Qatar will have even more advantages over competition. D, on the next page. Core screening is just one advantage we can offer with core screening is just one advantage we can offer with COAT that competition cannot. 4. Separation of HB core antibody positive plasma provides the opportunity to more efficiently identify plasma donors with high levels of other hepatitis marker antibodies, e.g. cyclomegalovirus and HB surface antigen. Sorry, I think that might be HB surface antibody rather than antigen. 5. It may be possible to fractionate core positive plasma to concentrate. Later development in AIDS screening, for example, HTLV3, can be used to test for concentrate. Much of it can be salvaged. 6. Startup costs are already sunk and testing costs are not financially significant. The exclusion of a plasma allocation of $13.25 per litre is the real expense. 7. We may be able to make stronger claims concerning non-A, non-B hepatitis risk from core screened products. So those are the 7 reasons in favour of continuing the testing. The arguments for discontinuing were then set out, and I quote those now. 1. Cost. A. Implementation of HB core antibody testing has cost somewhat more than $100,000. Continuation of this testing will add approximately 30 cents to all litres procured. B. 15% of plasma tested has been HB core antibody positive. Exclusion of this plasma from coagulation products and quarantine of HBsAg positive AHF concentrates will have significant but indeterminate costs. 2. HBcAb is a marker for hepatitis B and a potential indicator of population groups at risk for AIDS. It is not an AIDS-specific test and will not identify all potential AIDS victims. Even with continued HB core antibody testing, we are at risk for product withdrawal due to AIDS contamination. 3. More specific AIDS screening tests will be available in the future. HTLV3 is assumed to be the causative agent for AIDS. Press announcements indicate that an HTLV3 antibody test will be available by October 1984. I pause there to say so that we will be looking at the screening tests shortly. It is contentious whether this target will be met for commercial availability in quantities approaching blood/plasma screening requirements, i.e. 1.5 million per month. Coagulation product derived from HTLV3 tested plasma could be commercially available 8 to 11 months following implementation of testing. 4. The cut-to-dry heat treat method may demonstrate inactivation of HTLV3. Use of HB core antibody negative plasma plus heat treatment of coagulation products would then be considered a belt and suspenders redundancy. 5. Exclusion of hepatitis B core antibody positive plasma from coagulation products should reduce the already low level of HB surface antibody, thereby potentially increasing the risk for transmission of hepatitis B virus. 6. Flexibility of plasma procurement operation is considerably reduced by the requirement for HB core antibody testing. Many suppliers to our industry are unable to comply or will not comply with procedures necessary for separation and control of tested plasma. 7. Identification, separation and isolation of hepatitis B core antibody tested plasma has placed a large burden on Qatar employees and staff. Blending of hepatitis B surface antibody positive and hepatitis B surface antibody negative fraction 2 to assure normal ISG will increase this burden. So those are the pros and cons set out. I will come back to the figures that are contained in a second. I would just note that the company agreed to the continuation of the testing programme in a meeting on the 25th of June 1984. The reference to that is at paragraph 202 of the written presentation.
The document that you're showing me, I think it was anticipating a meeting which took place on the 31st of May.
It is. I don't know why it was, but the decision was delayed until the 25th of June 1984. We can trace that through if necessary, but the overall point is that Qatar continued that programme.
I mean, just to fit in what had happened between the starting of testing for anti-HBC by Qatar on the 2nd of April and this document, which is the 25th of May, 84, is that on the 21st of April, there was a press, Gallo announced his discovery that HTLV3 was, he claimed, the cause of AIDS. And 2 days later, there was a press conference with the Secretary of State for Health to announce it, and to announce that tests would be forthcoming. Yes,
the somewhat optimistic statement on the 23rd of April 1984 by Secretary Margaret Heckler was that the tests would be available within 6 months. And that is why I think there is a reference in the document that we've just looked at to the tests being available in October. And scepticism is expressed, and justified scepticism, it turns out, about whether or not that target can be met if what Secretary Heckler meant was that these tests would be available across the country at the level that the industry required. That, in fact, didn't take place until the early and mid part of the following year, 1985, and we'll come on to that in due course. The decision then by Qatar is to continue the testing. The meeting on the 25th of June 1984 also contains an interesting detail that an insurance claim had been made for a sum for to cover the costs of the withdrawal of 16 lots of Co-8 and Co-9 the previous year. That's the product withdrawal that we mentioned earlier. The insurance claim was for $4.9 million. That was for 16 lots of the products. If we could just go back, Shumik, please, to the second page of the document that we were just looking at, CGRA 40362. And if we could go to 1D. Now, this is the cutter rendering of the CDC position as of the time of this document, which is May 1984. There it is saying that the CDC reports 88% of homosexuals and bisexuals and 96% of intravenous drug users with AIDS are hepatitis B core antibody positive. Now, that contrasts with the figures given by Dr. Everett in 1982, which were, and I'll just read them to you rather than putting them up on the screen. The figure for homosexuals and bisexuals with AIDS in 1982 was 88.2%, so a very similar figure to the one that is contained in that document. The figure for intravenous drug users with AIDS in 1982 was 100%, so all 21 such cases. That has fallen slightly, but it's still very high, 96% as of May 1984. And the figure given in 1982 for probable AIDS, and the test then was lymphadenopathy, so swollen lymph nodes, was 81.3%, a very slight decrease by 1984 of 78% of individuals with AIDS prodrome are positive. That might not be comparing precisely like with like, but the figures you may feel are strikingly similar.
Yes, I mean, for low numbers, you'd speculate that they would be bound to be within the same confidence interval. Speaking statistically.
I wouldn't disagree with that at all. Those figures are notably different from the ones that were cited at the meeting that we have just looked at. I won't take you back over those. What I would add here is that according to the Creever report, and it's electronic page 772 of that report, I won't take you to it, but Mr. Justice Creever said that in early 1985, the CDC published a study that demonstrated that 62% of donors to whom CDC had traced a transfusion-related AIDS case had tested anti-HBC positive. So that figure of 62% seems to relate to all AIDS cases. And as we saw earlier, the correlation between anti-HBC positivity and the Haitian community was lower than that with the
well, it's looking at something a bit different there, isn't it? Because that's looking at donors. Whereas I think what you've just been showing me has been looking at cases.
Yes, that's correct. Yes.
So they're two different communities.
Two different. Those are donors to whom AIDS cases have been traced.
Yes.
But yes, they are a different community, and it would include donors who might fall outside the groups of intravenous drug users and homosexuals. But I stress that study had not been reported publicly at the time of this meeting. You may also feel, sir, that it is unlikely that the CDC would have made their figures available only to Qatar. There would have been no purpose of them doing so. Moving on from that meeting, and as you said, sir, but between those two meetings, you have the announcement from Dr. Gallo and then Secretary Heckler about the isolation of HTLV-3. By June 1984, 49 cases of AIDS in people with haemophilia had been identified in the United States. The following month, on 13th of July 1984, there was a publication in MMWR, which was a preliminary report prepared by the CDC using the first generation of LAV tests, so the tests that they had developed with Dr. Montagna. And that showed that 18 of 25 asymptomatic people with haemophilia tested positive for LAV, as did 2/3 of a larger sample size. The references for those are at paragraph 205, written presentation. On the 16th of July, the study group that had been chaired by Dr. Riddell on anti-HBC testing presented its final report. The majority continued to oppose the introduction of the test. They cited the lack of specificity, the 6 to 20% rejection rate for plasma, an increased risk of hepatitis B transmission, plasma shortages, and price increases. The minority argued that the tests would have identified 60 to 80% of homosexual men, but they accepted that it would be they accepted that it would no longer be worth implementing anti-hepatitis B core testing in light of the identification of HDLV3 and the prospect of a screening test that was specific for AIDS. Later that month, July 1984, there was some discussion of an industry-wide public relations campaign. You've seen the documents in respect of that, sir. We don't know how far that campaign went. The references are at paragraph 207 of the written presentation if people wish to pick them up. So that takes us to the end of July 1984, which is where we're going to break with the strict narrative, chronological narrative that we've had so far. To summarise the position at that point, the debate about the safety of blood products in the first half of 1984 had predominantly been about anti-hepatitis B core testing. A coalition of two of the fractionation companies, ARMA and Alpha, and a number of blood banking organisations had successfully resisted calls from the FDA to introduce such testing, and had done so until a point in time when even advocates of testing were resigned to waiting for the emergence of a specific test for HDLV3. However, two companies, Highland and Cutter, had taken a contrary view, and the latter had unilaterally introduced anti-HBC screening tests, despite pressure not to do so. Product withdrawals had continued in the first 6 months of 1984, as had the increased number of AIDS cases identified among people with haemophilia. Initial results on patient testing had also provided evidence of devastating rates of infection within that community in the United States.
Do we actually know how many withdrawals there were in this part?
Up until this point, 4.
In 1984?
Yes. So,
they're the 4 you mentioned earlier?
The 4 that we mentioned earlier, yes.
So when you say product withdrawals had continued?
That they had continued, the last 2 of those withdrawals had been in the early months of 1984.
Thank you.
The year had, however, seen the announcement of the isolation of the HDLV3 virus and the granting of licenses for heat-treated products. All 4 companies had a license for heat-treated products, and it's to heat treatment that we will now turn. In the written presentation, I set out why it is that no effort is made today to try to provide a comprehensive presentation on the development of heat treatment. It is a very complicated and dense story. The main narrative events are the ones that I will seek to highlight today. There is an abundance of evidence, you may feel, sir, that during the 1970s, factor 8 and factor 9 were considered to be highly heat-labile proteins, such that they would not survive the sort of heat treatment that was used as a method of viral inactivation on, for example, albumin. And you will recall that the evidence of Sue Middleton was good. However, in 1977, the German company Behringwerk commenced work on heat treatment studies with a view to providing factor concentrates with lower risk of hepatitis transmissions. That's 1977 that we have a date for Behringwerk beginning these studies. By the following year, 1978, Highland had begun some form of research on heat treatment of haemophil. There is a memorandum dated the 26th of April 1978 stating that that work required additional funding and manpower if it was going to be progressed. Highland's parent company, Travenol, appears to have obtained information from its German subsidiary about the Behringwerk process. That encouraged further efforts in that field. If we could have on screen, please, CGRA 40222. This is a internal Highland document marked Confidential Division Priority List, September 1979. The division I take to be Highland Division of Travenol. One, top of the priority list, is Autoplex, which was the product intended for patients with factor VIII inhibitors. And two, anti-hemophilic factor hepatitis risk removal. A, heat inactivation stroke radiation, and B, ultrafiltration and glycine. So two different methods of viral inactivation, which are the second item on Highland's division priority list as of September 1979. There are other documents are referred to in the written presentation as well. Behringwerk obtained a German license for its product in early 1981. That product was pasteurized, so that's heated in a solution, at 60 degrees centigrade for 10 hours. Important throughout this section to keep in mind the different methods of heat treatment as well as the different temperatures used. And Behringwerk's was a true pasteurization technique, which means that the product was heated in a solution. That product was not at that time licensed or marketed in the US and the UK, and there were various drawbacks to it, including the fact that there was very low yield. At about the same time, early 1981, Highland were proceeding to clinical trials in humans of haemophil T. We heard a little about that from the evidence of Dr. Kingdon that we looked at in September and October. Other companies had also commenced work on heat-treated products by this time. There is a Qatar budgeting document from 1983. I won't bring up the document, but I will just summarise what we have taken from it, and it's at paragraph 215 of a written presentation. That document suggests that in 1981, the total amount spent on research and development by the Drug Research and Development Division was $4.2 million, or $4.27 million. And of that, $293,000, 6.9% of the budget, was spent on hepatitis-free product development. In 1982, an estimated $558,000 of the same budget was forecast to be spent on that project, which was 9.2% of the total. In addition, an estimated $102,000 was estimated to be spent from the Technical Operations Division, on the project, which was 5.1% of that budget. The notes to the budget describe the project as being, and I quote, a defensive move necessary to maintain AHF market share. The 1983 budget included a projected $632,000 under the international division budget, which was 36% of that total budget, which seemed to be earmarked for applications for a Japanese license and for testing in the first quarter of 1983. Now, we don't suggest that these numbers are the sole figures for what was spent by that company at that time. They're what we can discern from the document that we have. If we could have on screen, please, Shumik, JREE.
Well, just before you do that, this is looking at factor 8.
The project is referred to as hepatitis-free product development.
Because I have a note to myself that the reference is BAYP 303708, that the Qatar's Biochemical Research Department quarterly progress report for April to June 1972, so 10 years earlier, more or less, showed that experiments had been conducted to see if virus with which factor 9 had been spiked could be inactivated without the factor 9 losing its activity. And it suggested that the activity was indeed maintained if certain salts were used. In other words, that factor 9 could be stabilized. And I think the virus in that experiment remained active. But it may indicate that there were certainly moves in the early 70s to look at whether heat and stabilizers might be an answer, as far as Qatar was concerned.
Yes, I think it's fair to say that the heat development techniques that were developed in the early 1980s were generally applied to both factor 8 and factor 9, but there were additional difficulties with factor 9, particularly in establishing that the resulting product didn't cause blood clots. And we heard a little about that from Dr. Middleton. There was a considerable concern amongst the clinicians about thrombogenicity.
Yes.
But the way, when we looked at the licensing of these products, we would often see that the same heat treatment regime was applied both to factor 8 and factor 9. And so I have taken it that the work in the early 80s was being done generally on factor concentrates, and then the licenses would be applied for specifically for the factor 8 and factor 9.
I think my question was directed towards activity on the research front anyway during the 1970s, given that there had been some evidence of some in 1972.
Yes, as I understand it, the concern in the 1970s, and it's often in the papers presented as almost a received wisdom.
I mean, do we actually have any documents about this that you're aware of so far as Pharma are concerned?
But there are many thousands of documents that we have, and there will be some references to, or there are certainly from Qatar, there are a large number of documents in which do refer to research products.
Because in due course, I will be quite interested in the progress of research into inactivation of virus in products during the 1970s.
That is something that we can look into, sir. It is
but
fairly significant.
This is now, I think, not the time to to deal with it.
I certainly won't be able to do it on an extempore basis. If we could have JREE 5019 on the screen, please. Page 106. This is taken from the Institute of Medicine report. It's a helpful table which sets out when licenses were applied for and when they were granted. I've mentioned these as we went through, but it's perhaps helpful just to look at this now. Baxter, which is the parent company of Highland, the application for a license was made in June 1982, and it was granted in March 1983, a period of 9 months. We then have Miles, which is, perhaps I think it might be more helpful actually to take it chronologically. So Baxter, June 1982, granted March 1983. Armour, this is Factor 8, December 1982, granted January 1984, so 13 months. Alpha, which is, I should have mentioned, the Factor 8 is dry heated. The Highland product was dry heated. There was a different, both were heated at the same temperature, 60 degrees. Hemophil was heated for 72 to 74 hours, and Factor 8 was heated for 30 hours. That may be of some significance later. Alpha applied for its license for Propylate in December 1982, and it was granted in February 1984. It's referred to as wet heat there. As we've discussed before, sir, it was heated in suspension, so not a true pasteurization, but a suspension for 60 degrees centigrade for 20 hours. And then we have Cutter. They applied for and received 2 licenses. The first was for a pasteurized product, heated 60 degrees C for 10 hours, so similar to the Behringwirth product. That's applied for in August 1983 and approved in January 1984. And then a dry heated product, 68 degrees centigrade for 72 hours, applied for in November 1983 and granted in February 1984. So the speed at which these licenses are granted increases in late 1983. I also point out there that the Miles dry heat treatment was heated at a higher temperature, 68 degrees, than both the Highland and the Armour product, and it was heated for 72 hours, which was longer than the Armour product and about the same time as the Highland product. Immuno don't appear to have sought an FDA license for their heat-treated cryobalin. Their UK application was made in December 1984, and it was approved in February 1985. So a short period, and you will recall, sir, from the licensing section, that the DHSS were encouraging the pharmaceutical firms in the autumn of 1984 to put in applications for their heat-treated products.
When the meeting at Heathrow Airport took place on the 24th of January 1983, Immuno were presenting on a method of chemical inactivation.
Yes, they were.
Do we know anything more about that? Did anything come of it?
There are a couple of documents from later in that year where they...
Just take a moment. Yes.
There were a couple of documents from later in that year which indicated that Immuno had given some presentations at conferences hinting at this process and the work that they were doing on it. What appears to have happened or may have happened is that the heat-treated products were seen as a more likely avenue of success from later in 1983, and therefore Immuno's attention turned towards that method and away from the chemical separation that they had been previously discussing. I have to say the chain of documents doesn't allow for a full answer to that question. That is my analysis of what was going on. And it's a pertinent point, so because although we are focusing here on heat treatment, as we saw with the earlier Highland document, there were other methods of viral inactivation which had been considered and worked upon by the companies as well. But by this stage, heat treatment seems to have been the one that had gained priority from all of the companies. The New York Blood Center had worked on a solvent detergent method in the 70s and the 80s, and that was later picked up by some of the companies and taken forward. That led to the second generation of products which were safer in respect of non-A, non-B hepatitis in particular. It's clear from the documents and indeed from the chronology that the initial work that was undertaken on heat treatment was for the purpose of inactivating hepatitis and not AIDS. It predated the knowledge of AIDS. And we've seen the meeting that took place on the 9th of September 1982, where fractionators met with officials of the US federal government, and the discussion there was on hepatitis, and AIDS wasn't even mentioned in the record of the meeting. I won't go back to that meeting, but at paragraph 219 of the written presentation, there is a summary of some of the topics that were discussed and some of the difficulties that the companies were having as of the 9th of September 1982 in heat treating their products. Difficulties in plasma protein denaturation, the protein being heat labile, difficulties with chimpanzee experiments, which were described as being slow and fraught with inherent problems, whether to change inactivation processes known to inactivate hepatitis B virus in order to seek to inactivate non-A, non-B hepatitis, and the use of marker viruses for non-A, non-B hepatitis and the difficulties involved in using them. No consensus could be reached on that point. Non-A, non-B at that stage was known to exist, but there was no test for it, which hampered an ability to assess how effective your product was. And there was also the discussion about the use of clinical trials, which including clinical trials following on from chimpanzee trials. So those were some of the difficulties that were present in September 1982. Turning back then to the period from 1983 and 1984 when the products began to come on the market, the early heat-treated products were treated with some skepticism by haemophilia clinicians. There were doubts, which turned out to be justified, about their effectiveness in inactivating hepatitis, and particularly non-A, non-B hepatitis. And doctors were concerned that heat treatment of the products could give rise to the development of inhibitors in patients. And again, Dr. Middleton mentioned that in her evidence. Dr. Everett, in his tragic history, and I won't take you to it, but I quote him from it, wrote that, quotes, because of the high cost and feared risk, the Highland product did not achieve widespread use. Dr. Kingdon gave similar evidence in his draft witness statement from 1990. So there was a reluctance from some haemophilia doctors to take on these heat-treated products. Just to re-emphasize, the three areas that would emerge from the evidence are questions about their effectiveness against hepatitis, concerns that they may give rise to the development of inhibitors, which would mean that patients could no longer be treated with standard factor VIII concentrates, be they heat treated or not, and also the factor of cost. These products were more expensive. Similar doubts were expressed in the United Kingdom, and I've given some references in the written presentation to, for example, a letter that Dr. Gunson wrote to Dr. Walford on the 29th of June 1983. And there was also a discussion of heat-treated products on the 13th of July 1983 meeting of the Biologicals Subcommittee of the Committee on the Safety of Medicines. As late as June 1984, Dr. Aladort, who was by then working for the World Federation of Haemophilia, or at least working with the World Federation of Haemophilia, wrote, and I quote, that he strongly opposed the industry's inclination to stop manufacturing non-heat-treated factor 8 and 9 concentrates. He cites the possibility of neoantigen formation with heat-treated products, the extra cost involved, and the fact that there is no direct evidence that heat treatment eliminates or reduces the risk of AIDS to haemophiliacs. So that's June 1984. Although the products were originally developed to tackle hepatitis, it was the AIDS crisis that led to them becoming more rapidly licensed and accepted for use. As we've seen from that table, the time lapse between an application for the license from the FDA and the license being granted shortened considerably as time passed, and we've mentioned as well the UK position in that respect. The work done by the CDC on developing an assay to measure viral load in factor concentrates was also an important catalyst. And that goes back to the cooperation that I mentioned before the break between the CDC and Dr. Montagna and his team. According to Dr. Evett in his tragic history, in August 1984, the CDC presented results of studies at a conference of the World Haemophilia Congress in Rio de Janeiro, which showed that there was LAV inactivation by heat treatment. And of course, by August 1984, HTLV3 had been isolated as well. There was some debate about whether or not the two were the same virus, but an increasing acceptance that they were. So that's August 1984 when those results are presented. According to Dr. Evett, clinicians still expressed scepticism, and that led to further experiments taking place, particularly conducted by Dr. Steve McDougall, Dr. Evett's colleague at the CDC. And Cutter, and later Alpha, were to provide concentrate to Dr. McDougall. Concentrate was then spiked with virus, subjected to the heat inactivation techniques in a laboratory setting, and the CDC demonstrated there that there was a reduction in viral inactivation. And that was taking place in September 1984. Also, in September 1984, Dr. J. Levy published results of his work on inactivating mouse retrovirus by heat treatment, and that was reported in The Lancet. And mouse retrovirus was thought to act in a similar way to retroviruses in humans. So gathering evidence as of September 1984 for the effectiveness of heat treatment. Events then moved rapidly. In September 1984, a conference was held between haemophilia clinicians and the CDC, at which there was agreement that physicians should consider changing to heat-treated factor concentrates. In October 1984, the Medical and Scientific Advisory Committee of the National Haemophilia Foundation advised physicians to, quote, strongly consider changing to heat-treated products. The CDC published the results of Dr. McDougall's experiments in MMWR on the 26th of October 1984, but of course preliminary results have been discussed with the various bodies before then. The same edition of MMWR recorded 52 cases of AIDS in people with haemophilia. The Groningen Conference of various haemophilia clinicians and fractionators took place on the 1st and 2nd of November 1984. Further presentations made there on the efficacy of heat treatment. In December 1984, an editorial in The Lancet advised that it was reasonable to take a pragmatic approach and switch haemophilia A patients to heat-treated products, notwithstanding the lack of clinical evidence of their efficacy against AIDS. So until that stage, the studies had been done on a laboratory basis showing viral inactivation. They hadn't been traced clinically, but despite that, The Lancet were saying that a pragmatic approach should be taken. In the same editorial and in the context of a call for additional funding to purchase such products, the editorial suggested that it would be, and I quote, indefensible to allow prescription and home use of material known to be at risk of HTLV-3 when apparently safer preparations are available. That is December 1984. In February 1985, the first published reports appeared in The Lancet again, showing the results of a clinical trial of haemophil T, which appeared to demonstrate that it did not transmit HTLV-3 to the patients that had used it. We heard something of that from Dr. Kingdon in September and October. That was Professor Manucci's trial. And Dr. Kingdon points out that it was the first clinical evidence of the efficacy of heat treatment to go with the laboratory evidence which had been emerging since August 1984. Dr. Kingdon said that Highland ceased the sale of all non-heat treated factor VIII concentrates in the United States in January 1985. And on the 13th of March 1985, Cutter wrote to Dr. Geoffrey Savage at St. Thomas's Hospital to say that the company, quotes, felt that it was prudent to no longer affect sales of non-heat treated product for use in the United Kingdom. From the 15th to the 19th of April 1985, a conference was held by the World Health Organization in Atlanta. And during the conference, the position that non-heat treated products should not be used was advocated, but it's not clear from the documents whether that generated a consensus of opinion. The conference, however, did endorse and recommend the use of heat-treated products. So there is a distinction between encouraging the use of heat-treated products and the question of whether you should also stop the use of non-heat-treated products. Also in April 1985, the National Haemophilia Foundation revised its treatment recommendations to recognise that heat-treated concentrates, quotes, may be the preferred products for infants and children with severe haemophilia and for newly identified and previously untreated severe haemophilia patients. It continued to advise the use of desmopressin or cryoprecipitate in patients with mild or moderate haemophilia, and it wasn't until December 1985 that the NHF advised that only heat-treated products should be available for all patients. That's all patients with severe haemophilia. So as well as these clinical recommendations which are going to the clinicians, the availability and use of heat-treated products and their apparent efficacy led to discussions about whether or not there should be a change in approach to the recall of products that were known to have been contributed to by a patient who later developed AIDS. In April 1985, so at around the time of the World Health Organization conference, the National Haemophilia Foundation reversed its previous position and declared that it no longer recommended the recall of concentrates contributed to by a donor later found to have AIDS if those concentrates have been treated with a licensed heat-treated process. The basis for that change in position is that the heat treatment would have, and I quote, adequately killed, that's the end of the quote, the virus contained in those concentrates. The change in the NHF's position was cited by Alpha in a letter to the FDA on the 30th of April 1985, in which it explained that it didn't intend to withdraw vials of factor concentrate that had been produced using plasma from a donor later diagnosed with AIDS. And that letter sets out a series of calculations in which the company shows that even on a worst-case scenario, the heat treatment process would, in their view, have inactivated the amount of virus that was in that product. And so they say there is no need then to recall it. And they also point to the fact that the NHF are no longer calling for such products to be withdrawn. The letter makes it clear that the company had discussed this position with an official of the Bureau of Biologics, and Counsel to the inquiry haven't identified any subsequent correspondence from the FDA saying, no, you shouldn't do this. While this was going on, non-heat-treated concentrates that were still in circulation continued to be withdrawn by companies. On the 3rd of May 1985, Armour withdrew 7 batches. And in June 1985, Highland announced a withdrawal of products. These are non-heat-treated products that were still available.
Was that in the States or here?
Those withdrawals were they were announced in the States. It's not always clear from the documents where the concentrate had gone to. We will be hearing tomorrow about an Armour withdrawal that specifically a product in the United Kingdom, and I'll refer to that a little later, but I'm not going to preempt the evidence, so I won't go into that in great detail. This was then a period where heat-treated product was becoming increasingly available and increasingly used, but non-heat-treated product was still in circulation. And there is some evidence that Qatar sought to sell non-heat-treated products, including from stock held in Britain, to overseas markets that had not at that time adopted heat-treated products. If we could have on screen, please, CGRA 40583. This is an internal memorandum from Qatar dated the 10th of April 1985. And it is entitled Inventory of Non-Heat-Treated Coate in the United Kingdom. And I'll read from the memorandum. In a recent trip report from Jack Wood, Jack indicated that the transfer price value in US dollars of the non-heat-treated coate inventoried in the United Kingdom is $395,273.53. As you know, we are attempting to sell some of this merchandise into Hong Kong and Taiwan. Jack indicated that Brian Dios will be discussing with Trevor Barracliff a dating extension allowing for relabel and continuous selling into markets not yet converted to the heat-treated Factor 8 products. I'll keep you posted as we progress in this project. Jack Wood was a Qatar employee who was involved in distributing Qatar products in Europe. And Trevor Barrowcliffe is a civil servant within the National Institute of Biological Standards and Control, NIBSC. Now, we don't know at present how far those discussions with Mr. Barrowcliff got, or Dr. Barrowcliff got. If we could go, please, to CGRA 40581. You can see that this is a memorandum from Jack Wood, who was referred to in the previous memorandum, dated the 15th of April 1985. And it's called Coate Returns from United Kingdom. We could just read this. It's addressed to a colleague called Gary. Gary, on behalf of Qatar UK, I would like to express our appreciation for the efforts you have made in marketing to other countries their inventory of non-heat treated Coate. The 4,000 vials of 250 material will significantly help in reducing this inventory, but we still have on hand close to 4 million units as listed in your letter. Two major lots are 2,877 vials of product reference number is given, and 1,434 vials of another lot of product. We will do everything possible to sell this product in European countries not yet converted to heat-treated product. Thank you again for your efforts, and we look forward to the positive results that you will achieve on the rest of this inventory. We can take that down. A similar memorandum was sent, which is referred to at paragraph 233 of a written presentation to another Qatar employee. And that memorandum referred to the fact that Qatar would be willing to subsidise the sale of the remaining non-heat treated product on the basis that it was better to have a lower profit than a total loss. And such an approach is also evident within the United Kingdom in this period. If we could have on screen, please, CGRA 40561. We can see that this is sent to Dr. Tuddenham at the Royal Free Hospital, and it's sent by Linda Frith, the Sales Development Manager at Qatar UK. And it says this: Dear Dr. Tuddenham, Brian Dios asked if I would contact you regarding a lot of Coate regular product in our inventory. We have available a lot of 1,045 vials, 260 international units, with an expiry date of the 13th of July 1985. After speaking with our financial advisors, I would like to lower the price of this material to 4.75 pence per international unit. I hope this price is more acceptable to you. If you are interested in this batch, could you let me know as soon as possible? Now, so we don't know whether or not this was heat-treated or non-heat-treated product, but the expiry date, the 13th of July 1985, the fact that this letter was being sent in May 1985 and in the context of a previous memorandum that we've looked at, and the very low price of the product, 4.75 pence per unit, might indicate that this is non-heat-treated product which is being offered on a discount basis. We don't know what Dr. Tuddenham said in response. A later memorandum dating from 1987 recorded that Qatar sold both heat-treated and non-heat-treated products in parallel in the United Kingdom until October of 1985. So that memorandum would certainly allow for the sale of non-heat-treated products within the UK as of May 1985. A formal letter that was subsequently sent by the company to a solicitor's firm, so one can infer that was part of legal proceedings, stated that after February 1985, non-heat-treated product was supplied, and I quote, only in response to specific requests for it from haemophilia centres. So I'm about to move on to a a meeting which took place in the United States in May 1985 on the question about what, if any, what regulatory action should be taken over non-heat treated products. That's the last sections that were really on heat treatment, and it will take a little bit of time to go through, so I wonder if now might be a...
Well, let's take a break then, shall we, and come back at 2 o'clock.